molecule that we're going to look at a little bit more in depth are our proteins. So here's just some simple facts about proteins. They are grouped, typo there, they are grouped according to their function. Some are structural, some do storage, transport, defensive, and enzymes. Those are just some major categories of proteins. The function of proteins is diverse, but their structure is very similar. And proteins are all polymers of their monomer, which is an amino acid. And we're going to look at these monomers between the amino acids are called peptide bonds. When we were in lab yesterday and you did the burette solution, what that indicator was looking for and was indicating for was the presence of a peptide bond. This slide here is just going to show you some of the protein functions. So just hint, be able to look at these and be able to recognize and identify what a function and what proteins do in your body. So let's look more carefully at the structure of an amino acid. Again, our amino acid is our monomer of our protein. So an amino acid is just a small organic compound. And we started this whole week by looking at functional groups. So if you look carefully, you're going to see here's our carbon chain, or really our central carbon. You have an amino group, which is your NH2 attached on one side, and on the other side, you have your carboxyl group. The most simple amino acid that you can have is instead, again, this R is just representing the things that you can put in that place. And so just by putting an H there, that's going to be glycine. That is your simplest amino acid. Again, because you have that R group and you can attach those side groups, and this is called the alpha carbon. Okay, it's just that central carbon. Those R groups are what's going to determine the uniqueness of the amino acid. That's what makes your proteins complex, because that side group can range from an extra carbon to carbon rings. What's really crazy when you think about it, though, all proteins are composed of the same 20 amino acids. So 20 different amino acids can be built. And so here are just some of the structure of some of the different amino acids. If you look more carefully, you should be able to identify what makes a nonpolar or a polar amino acid based upon what functional group or side group is attached to that alpha carbon. Now, when you think about your body and what your body can make, you have non-essential amino acids and you have essential amino acids. Non-essential amino acids can actually be synthesized or made by your body, but essential amino acids, they cannot be synthesized by your body. And you have to obtain those from the protein in your diet by eating protein. Only 9 of the 20 amino acids are non-essential, which means they can be synthesized in your body. What's interesting is you can have what's called a conditionally essential amino acid, and that's just when one of those amino acids that your body can make suddenly becomes essential because there's a mistake that occurs and your body is no longer synthesizing it. And PKU is an example of that. I'm not sure exactly how to pronounce it. I think it's phenylketonuria, but that is basically a disease where phenylalanine is not able to be converted into tyrosine. And so again, that's going to cause all kind of complications. As we look more carefully here, it is crazy how complex your amino acids and the building of the amino acids for the proteins is, and what happens if just one of those amino acids is sequenced incorrectly. So how do you get your amino acids to connect together to create a protein? Well, it's this peptide bond. And your peptide bond, if you look carefully, again, you're going to see your carboxyl group, and you're going to see the OH on the end of that functional group, carboxyl. And you're going to look at the other side of the amino acid, and you're going to see the amino group, and you're going to see the H there. And it's the same process that we looked at through the process of carbohydrates and dehydration synthesis. They are going to attach with the removal of water, so that nitrogen is going to be attached directly to that carbon, and that creates that peptide bond. A polypeptide is just a chain of those amino acids that becomes linked by that peptide bond. Now, this does require the input of energy, and the protein is just going to have that repeating pattern of your amino acid, your carboxyl group, your amino acid, I'm sorry, your amino group, your carbon, your carboxylic acid, your amino group, your carbon, your carboxylic acid group. Okay, that carboxyl group is also known as carboxylic acid. And what's going to be different about that and make it distinct are those R chains, those side chains. Just to look a little bit more carefully at how these amino acids link up, the amino acid, when it's in one of these polypeptide chains, is called a residue. So you can see here, I outlined the peptide bond. Again, the C to N linkage that gives off water. Here are your two peptide bonds, and so you have your amino acids. You're going to always sequence it so that your amino end is the start. Okay, that's the beginning. And we call these single amino acids that are linked together in this protein by these polypeptide bonds, we call it a residue. So you're going to have that repeating sequence of your NH2, your C, your CO double bonded attached to Na and N, your C attached to your double bond, and it's just going to keep going. So again, this is your residue, your single amino acid, and you're always going to sequence it starting with the amino side of that amino acid. Okay, that's how it's going to be written. Just want to kind of bring this to your attention, so this is important, hint, hint. When we have, and we're going to be looking at kind of the structure of some of the amino acids and how these, I'm sorry, not amino acids, of proteins and how they fold and what their structure looks like. And so sometimes they can fold in on each other and they create what are called disulfide bonds. This can only form between cysteine amino acids, and part of it is because they have a sulfur. And so again, disulfide means two sulfurs. So you're going to create that disulfide bond. So you take that amino acid, cysteine, cysteine, I guess you say it, and it creates the protein cysteine. There are no other amino acids that can do this because there is no sulfur to create that disulfide bond. So just be able to recognize that cysteine protein. The cysteine protein, again, is created by the disulfide bond. And I'm going to add this video later in the Google Forms so you can just look at it a little bit more and see how that disulfide bond actually forms. Now again, structure determines function. So the shape of our protein, so once we link all of these amino acids up together through this peptide bond, we have the possibility to form some different three-dimensional shapes. So the simplest shape of our protein structure is our primary structure. And this is basically a chain. And the order, the way we look at this is we look at the order of amino acids, and it's going to use those three letters to represent the amino acid. And I'm pretty sure you did this in biology. You sequenced for them. You use that codon chart to figure out start and stop and UAA and all of those genetic codes. You figured how they would code for the protein. And so when you just have this unique sequence and number of amino acids, and it's in a polypeptide chain, this is your primary structure. The unique sequence, again, for every protein is ultimately determined by whatever gene is encoding that protein. And so if you have a mistake in that gene sequence and you code for the wrong three-letter coding for that protein or for that amino acid, it's going to cause a different amino acid to be added to that peptide chain. And that's going to cause a change in the structure and function of that protein. So that is the simplest level of protein structure. Your primary structure, straight chain, amino acids coded by your genes. The second is your secondary structure. And this is just, again, this is a three-dimensional shape. And what's going to happen is if you have amino acids, you're going to have attraction hydrogen bonding that's going to occur between your amino and carboxyl group, and they're going to kind of bend upon each other. And they can create either an alpha helix, which is your spiral, or a beta pleated sheet, which looks like a folded plane. The third structure is called the tertiary structure. And this is going to include more of a three-dimensional shape. And you can see, again, so we have these R chains that can have different compounds and functional groups that attach to the R chain. So you're going to have different attractions. So you might have some hydrogen bonding occurring. You might have some more disulfide bonding occurring. You're going to have hydrophobic interactions where you have nonpolar and nonpolar attraction. You may even have some charges that are happening where your ionic bonds are occurring. And this is going to create, again, that globular shape as it goes from bending and folding in those spirals to more of a three-dimensional blob, basically. And then finally, when you take multiple blobs, that's when you have a protein that's assembled from two or more separate polypeptide chains. And so you have, again, multiple tertiary structures that come together to make that quaternary structure. I'm going to throw this video in from the Amoeba Sisters as well into today's Google form, just so you can watch it. They do a really good job. Again, they can use graphics and animation, where I'm just kind of using a still slide to explain how that structure and folding occurs. So what are your responsibilities? Please make sure you've reviewed slides 44 to 48. I've also added some additional readings and videos so that you want to make sure that you know what, is it domain? I forget what the word is. I'm going to look it up and I'll put that note in Google, the Google form, and I'll say, hey, make sure you know what this is. And then please make sure you've reviewed nucleic acids. So if you have questions, I'm going to be here in some office hours on Teams. Please jump out and ask questions. Make sure you're looking at those readings and videos. And remember, you have 3 attempts to go through those quizzes. Take it multiple times, get your best score.